Dear viewers, this video is part 3 in the 6-part series on the cell and its functions in Medicine for Common Man series. Lysosomes. See figure 1.3 shown earlier for lysosomes. Lysosomes shown in figure are vesicular organelles that form by breaking off from the Golgi apparatus. They then disperse throughout the cytoplasm. The lysosomes provide an intracellular digestive enzymes that allows the cell to digest the following: 1. Damaged cellular structures, food particles that have been ingested by the cell, and unwanted matter such as bacteria. Lysosomes are different in various cell types but are usually 250 to 750 nanometers in diameter. They are surrounded by typical lipid bilayer membranes and are filled with large numbers of small granules 5 to 8 nanometers in diameter, which are protein aggregates of as many as 40 different hydrolyses, that is, digestive enzymes. A hydrolytic enzyme is capable of splitting an organic compound into 2 or more parts by combining hydrogen from a water molecule with one part of the compound and combining the hydroxyl portion of the water molecule with the other part of the compound. For example, protein is hydrolyzed to form amino acids, glycogen is hydrolyzed to form glucose, and lipids are hydrolyzed to form fatty acids and glycerol. Hydrolytic enzymes are highly concentrated in lysosomes. Ordinarily, the membrane surrounding the lysosome prevents the enclosed hydrolytic enzymes from coming into contact with other substances in the cell and therefore prevents their digestive actions. However, some conditions of the cell break the membranes of lysosomes, allowing release of the digestive enzymes. These enzymes then split the organic substances with which they come in contact into small, highly diffusible substances such as amino acids and glucose. Some of the specific functions of lysosomes are discussed later. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Peroxisomes. Peroxisomes are physically similar to lysosomes, but they are different in two important ways. First, they are believed to be formed by self-replication or perhaps by budding off from the smooth endoplasmic reticulum rather than from the Golgi apparatus. Second, they contain oxidases rather than hydrolases. Several of the oxidases are capable of combining oxygen with hydrogen ions derived from different intracellular chemicals to form hydrogen peroxide. Hydrogen peroxide is a highly oxidizing substance and is used in association with catalase, another oxidase enzyme present in large quantities in peroxisomes, to oxidize many substances that might otherwise be poisonous to the cell. For example, about half the alcohol that a person drinks is detoxified into acetaldehyde by the peroxisomes of the liver cells in this manner. A major function of peroxisomes is to catabolize long-chain fatty acids. The figure 1.4 shows a pancreatic acinar cell with secretory granules. These vesicles store protein pre-enzymes. Secretory vesicles. See figure 1.4, secretory granules. One of the important functions of many cells is secretion of special chemical substances. Almost all such secretory substances are formed by the endoplasmic reticulum, Golgi apparatus system, and are then released from the Golgi apparatus into the cytoplasm in the form of storage vesicles called secretory vesicles or secretory granules. Figure shows typical secretory vesicles inside pancreatic acinar cells. These vesicles store protein proenzymes, that is, enzymes that are not yet activated. The proenzymes are secreted later through the outer cell membrane into the pancreatic duct and then into the duodenum, where they become activated and perform digestive functions on the food in the intestinal tract. This figure 1.5 shows a mitochondrion. This organelle extracts energy in the form of ATP, that is adenosine triphosphate, from nutrients. Mitochondria. See figure 1.5 shown earlier. The mitochondria shown in figure are called the powerhouses of the cell. Without them, cells would be unable to extract enough energy from the nutrients and essentially all cellular functions would cease. The morphological appearance of the mitochondrion is most intriguing. It's essentially a balloon inside a balloon. The outer membrane and inner membrane define two distinct internal compartments, the intermembrane space and the matrix space. The surfaces of the inner membrane is thrown into dramatic folds called cristae. This organelle is about 0.2 micrometer in diameter, which is at the limit of resolution of the light microscope. Many cellular reactions are also catalyzed within the mitochondrion. Mitochondria are present in all areas of each cell's cytoplasm, but the total number per cell varies from less than 100 up to several thousand, depending on the energy requirements of the cell. Cardiac muscle cells, called cardiomyocytes, for example, use large quantities of energy and have far more mitochondria than fat cells, that is adipocytes, which are much less active and use less energy. Furthermore, the mitochondria are concentrated in those portions of the cell responsible for the major share of its energy metabolism. They are also variable in size and shape. Some mitochondria are only a few hundred nanometers in diameter and are globular in shape, whereas others are elongated and are as large as 1 micrometer in diameter and 7 micrometers long. Still others are branching and filamentous. The basic structure of the mitochondrion, shown in figure, is composed mainly of two lipid bilayer protein membranes, an outer membrane and an inner membrane. Many infoldings of the inner membrane form shelves or tubules called cristae, onto which oxidative enzymes are attached. The cristae provide a large surface area for chemical reactions to occur. In addition, the inner cavity of the mitochondrion is filled with a matrix that contains large quantities of dissolved enzymes necessary for extracting energy from nutrients. These enzymes operate in association with oxidative enzymes on the cristae to cause oxidation of nutrients, thereby forming carbon dioxide and water, and at the same time releasing energy. The liberated energy is used to synthesize a high energy substance called adenosine triphosphate, shortly known as ATP. ATP is then transported out of the mitochondrion and diffuses throughout the cell to release its own energy wherever it is needed for performing cellular functions. Mitochondria are self-replicative, which means that one mitochondrion can form a second one, a third one, and so on, whenever the cell needs increased amounts of ATP. Indeed, the mitochondria contain DNA similar to that found in the cell nucleus. DNA is the basic constituent of the nucleus that controls the replication of the cell. The DNA of the mitochondrion plays a similar role controlling replication of the mitochondria. Cells that are faced with increased energy demands For example, in skeletal muscles subjected to chronic exercise training may increase the density of mitochondria to supply the additional energy required. Cell cytoskeleton, filament and tubular structures. See figure 1.3 shown earlier. The cell cytoskeleton is a network of fibrillar proteins organized into filaments or tubules. These originate as precursor proteins synthesized by ribosomes in the cytoplasm. The precursor molecules then polymerize to form filaments. As an example, large numbers of actin microfilaments frequently occur in the outer zone of the cytoplasm, called the ectoplasm, to form an elastic support for the cell membrane. Also, in muscle cells, actin and myosin filaments are organized into a special contractile machine that is the basis for muscular contraction. Intermediate filaments are generally strong, rope-like filaments that often work together with microtubules, providing strength and support for the fragile tubulin structures. They are called intermediate because their average diameter is between that of narrower actin microfilaments and wider myosin filaments found in muscle cells. Their functions are mainly mechanical and they are less dynamic than actin microfilaments or microtubules. All cells have intermediate filaments, although the protein subunits of these structures vary depending on the cell type. Specific intermediate filaments found in various cells include Descemet filaments in muscle cells, neurofilaments in neurons, and keratins in epithelial cells. A special type of stiff filament composed of polymerized tubulin molecules is used in all cells to construct strong tubular structures, the microtubules. Another example of microtubules is the tubular skeletal structure in the center of each cilium that radiates upward from the cell cytoplasm to the tip of the cilium. This structure is discussed later. Also, both the centrioles and mitotic spindles of cells undergoing mitosis are composed of stiff microtubules. A major function of microtubules is to act as a cytoskeleton, providing rigid physical structures for certain parts of cells. The cell cytoskeleton not only determines cell shape but also participates in cell division, allows cells to move, and to provide a track-like system that directs the movement of organelles in the cells. Microtubules serve as the conveyor belts for the intracellular transport of vesicles, granules, and organelles such as mitochondria. This figure 1.6 is the structure of nucleus. Nucleus has nuclear pores, nucleoplasm, nucleolus, and chromatin material. Nucleus. See figure 1.6, structure of nucleus shown earlier. The nucleus is the control center of the cell and sends messages to the cell to grow and mature, replicate, or die. Briefly, the nucleus contains large quantities of DNA which comprise the genes. The genes determine the characteristics of the cell's proteins, including the structural proteins, as well as the intracellular enzymes that control cytoplasmic and nuclear activities. The genes also control and promote cell reproduction. The genes first reproduce to create two identical sets of genes. Then the cell splits by a special process called mitosis to form two daughter cells, each of which receives one of the two sets of DNA genes. Unfortunately, the appearance of the nucleus under the microscope does not provide many clues to the mechanisms whereby the nucleus performs its control activities. Figure shows the light microscopic appearance of the interphase nucleus during the period between the mitosis, revealing darkly staining chromatin material throughout the cytoplasm, throughout the nucleoplasm. During mitosis, the chromatin material organizes in the form of highly structured chromosomes, which can then be easily identified using the light microscope. Nuclear membrane. The nucleus stores, replicates, and reads the cell's genetic information. The nucleus serves as a cell's repository for its complement of chromosomal DNA. To conceive of the nucleus as simply a hermetically sealed vault for genetic information, however, is a gross oversimplification. All of the machinery necessary to maintain, to copy, and to transcribe DNA is in the nucleus, which is the focus of all the cellular pathways that regulate gene expression and cell division. So, dear viewers, this is the end of part 3 in the 6-part series on the cell and its functions in Medicine for Common Man series. Dear viewers, thank you for watching the video.